special treat, as every time we do this is a special treat, but today we get to hear from Dr. Cathy Proenza. Cathy, but before she does, I just want to give a brief introduction about her. She got her PhD from Colorado State, so she's a Ram. I don't know if you root for the Rams or not, but no. Okay, good. She did a postdoctoral fellowship at Simon Fraser University in Canada, and also one at Harvard Medical School. And then she started on the faculty here in 2008. She was appointed as assistant professor, and last year she was promoted to associate professor. She also has a joint appointment with cardiology, and you'll understand why shortly as she gets into her science, which is fascinating for those of us who have had a chance to hear a little bit about it. As we were just chatting, and some of you got to hear, Cathy used to be a hotshot, which is a wildland, one of the elite wildland firefighter crew for. Do they work for BLM or Department of Interior or something like that?
Forest Service.
Forest Service, okay. So yeah, she's not just a scientist, she's also an avid outdoors woman who likes to climb rocks and backpack and ski and all sorts of, pretty much everything we do here in Colorado. She is expert. Is that Vestal Peak?
It's near there.
Okay.
It's a couple peaks over from that.
Okay, yeah, it looks like San Juan. So I'll turn it over to Cathy and she'll tell us about it. This is a science.
All right, so I put together some slides. Actually, I don't know all these guys. I don't know. I've seen, but I don't know you. Don. Okay. I know Meg. Okay. I don't know. Carol. Okay. And I know that. Okay. Hi, nice to meet you. Right, and you just moved in, right? Yeah, okay. Because I've seen you in the hall, but. Okay. I know Nancy.
And you know me.
So the work part has expanded.
Okay. Yeah. What I've done is I just, of course, kind of just pasted some slides that I have from other talks, and instead of trying to do this kind of comprehensive thing of blah, 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 what we do, I'm going to try to tell one story that sort of exemplifies kind of one aspect of our work. And my idea here is that this is supposed to be interactive, it's supposed to be learning. You guys have different backgrounds. If you're going to take me through, like, you guys spreadsheets, you have to go really slow. And I have to ask questions, and so I'd rather do it that way. I don't have an agenda here except to make sure that you kind of get sort of the gist of what we're doing and maybe learn a couple of things and not try to like go through a whole bunch of stuff and then you don't kind of really maybe get anything. Right. So yeah, we study cardiac pacemaker. And I do love mountains. So I'm gonna start with the heart, and this is I start all of my talks this way, and this is really why I do it. Okay, so here's the heart, and it can be all by itself. It doesn't need any neural input. So this is intrinsic property of the heart, and to me that's really fascinating. And then also most of the people in this department study the brains. Right. So what's going on? I'm a basic scientist. I'm going to tell you about sort of the translational aspects of like why does NIH, why should the taxpayers pay for this? Why should we care? But honestly, there's this other part of it. Is like this is a fundamental thing. One's heartbeat. How does that work? I think that's what really motivates me as a scientist is this sense of curiosity. And I think that's what I try to convey to the students, especially in large lecture classes and stuff like, is this going to be on the test? It's like, well, at some level, it's just kind of interesting. And it helps people to learn that way, to remember that way. All right, so the heart beats all by itself. And of course, we'll want to know why. So then we kind of whittle down. And it's been known for a long time, for more than 100 years, that the origin of the heartbeat is in this little area on the back right wall of the heart. It's called the sinoatrial node. So this is the natural natural pacemaker region of the heart. And the business end of it are these single cells. So they're sinoatrial node myocytes. So they're muscle cells, cardiac muscle cells, even though their job is to make electrical signals. I'll talk about that in a minute. So they're muscle cells, sinoatrial node myocytes. And this is a view, I think I showed you guys a few years ago when we did this before. This is a view through the microscope on one of our recording rigs, and so maybe two. So these single cells, isolated from mice, they just sit there and eat like for hours. And to me, this is kind of creepy and wonderful and fascinating and Halloween-y. Like, I don't know. They shouldn't do that somehow. And that's part of the curiosity. So this is kind of what the system is that we use. So these are hormones. And what makes this single cell go, what I just alluded to, is an electrical impulse, right? So even though the myofilaments, the muscle part of it conveys the action, what's triggering each of the little contractions in the cell is an electrical impulse, and that's what we measure. So I'll show you that. Oh, before I do that, I'm going to tell you why NIH works. What's the significance here? So ultimately, these cells are driving heart rate. We know that they're regulated by the autonomic nervous system to so that fight or flight response acts on these single cells. But sometimes the heart goes too slow and sometimes it goes too fast. And these can be really maladaptive for people. So if we have drugs to control heart rate, that can be a beneficial treatment for many kinds of heart disease. So in some cases, you want to decrease heart rate. So people with some kinds of heart disease benefit from a reduction in heart rate. So if the heart's beating really fast, the idea is that the heart muscle itself doesn't have much time for perfusion, so the blood can't kind of go through the muscles that comprise the heart. So if you slow it down a little bit, that can be helpful for people with some compromised cardiac function. And so right now there's one drug that is used to slow heart rate. It's called ivaridine. It acts on these ion channels I'm going to talk about. And it's beneficial for people, but it has some side effects, like a lot of first generation drugs. It acts on other things. And so we think that if we understand more about pacemaking, we could design better treatments, right? Including making a kind of a cleaner drug, say to slow heart rate. Then in the other direction, it's beneficial to increase heart rate, particularly for a pretty big population of older individuals who have sinoatrial node dysfunction, which means their pacemaker isn't working so well. And the only treatment we have currently are these implanted electronic pacemakers. These things are kind of big. They have batteries. They have these wires on them. They require surgery to implant. Most of the people who get them are like 85 years old. So surgery is kind of a bad idea. And it can be repeated surgeries. They're really expensive. There's a lot of limitations to using this kind of approach to increase heart rate for people. But it's beneficial. It keeps mostly improves quality of life. So people have fainting spells, or they can be kind of confused because their brain is not getting enough blood. So this is, pacemakers are really pretty well tolerated and pretty beneficial. But again, there's sort of room for improvement in treatment. So this is always the first part of my grants. And even though I want to know why these cells do that and how they work, the significance for human health is real. And it does help to know that I think what we're doing can have long-term benefits for people and for society. So that's my little sales pitch on that. Okay, so I'm going to go back to what I was just saying. These cells, I showed you that they sit there in the dish and they beat, and each beat is triggered by an electrical event. And so that's what I'll show you next.
Cathy, does the pacemaker hook to the sinoatrial node in the heart? Is that what it does?
It can, or it can patch onto other parts of the heart and kind of bypass the natural pacemaker. And there's different kinds of kind of pacemakers and ventricular assist devices and
My
stuff.
dad has a pacemaker.
Okay.
A pacemaker stimulator.
Oh,
What the difference is. It's on the second or third one.
Right. Yeah. So that's exactly the typical story, is that it's often requires kind of a lot of
But again,
different
the
devices.
defibrillator part of it too is.
Yeah, so the defibrillator, so when the heart beats, I'll try to go backwards. When the heart beats, I'm not sure you can see it too well on there. You might know there's different chambers on the heart. There's kind of these two atria up on the top, and then two ventricles on the bottom that are bigger. So the atria are these kind of little pumping chamber or priming chambers, and then the ventricles are the ones that are pumping the blood. One, the left ventricle pumps the blood through the whole body, and then the blood comes back, it's all deoxygenated, and then the right ventricle pumps the blood back through the lungs so it becomes oxygenated again. And so the defibrillators usually are in case there's a maladaptive electrical signal. So, then the pacemaker is maybe not triggering the natural sinoatrial node is not really triggering contraction. In order for this to work right, you have to have this really coordinated, this chamber pumps, and that one does, and the chambers have to fill before they can send the blood back out. And, it's a conceptually simple system, right? But the timing is really important. And so fibrillation is when the heart muscle just starts sort of contracting really rapidly, and it's not pumping efficiently. So if it's atrial fibrillation in those top chambers, that's associated with stroke in a lot of cases. Ventricular fibrillation, the heart's not pumping blood at all, and it's lethal. So a defibrillator catches that right away and starts the heart again. Does that answer that question?
Yeah. I knew we had both, and I didn't know how they work together.
Yeah, so it's going to be kind of a complicated thing. It's like, okay, we want the heart to go this pace, this rhythm, and then we want to make sure that the electrical signal spreads through the heart so that the pumping action is coordinated. So it's probably dealing with both parts of that, from a superficial view. And it's hard for me to answer some of these questions because I always have to say I am not a medical doctor.
Right.
I know about cells and ion channels and molecules and stuff. Yeah, and so I don't want to dispense any medical advice. Right? But that's great. So I like answering questions. More of a conversation. All right. So there's that. Okay. So here's another one of these cells. Another example of single cells. And here you can see this microelectrode. To drive the microelectrode. So here's this little microelectrode. So these are made out of glass capillary tubes. They're really small in diameter, and they're pulled to this very fine point. See a scale bar. So this is just like 1 micron or less of this microelectrode.
So that's what we would have used in the synapse lab, correct?
Maybe
we got to
use
sit through.
glass microelectrodes.
Yeah, they
for that.
use it to stimulate the muscle cell.
And the machine that hold it.
Yeah. Okay.
Yeah,
Exactly.
yeah.
That's exactly
Yes.
the
Okay.
same.
That
I wasn't
might
sure
be
what you guys.
Yeah.
Right. So we can make measurements from these single cells, and now this is plotting membrane voltage as a function of time, and you can see these spikes in membrane voltage. They're called action potentials. So neurons fire action potentials. This is the basis of all electrical signaling. And it turns out that muscle cells also are electrically excitable cells. So skeletal muscle cells and these cardiac muscle cells have these unique action potentials. So what's interesting about these action potentials, and pacemaker cells is they occur all by themselves in the same way as that whole heart doesn't need the brain to tell it to beat. These single cells just fire action potentials, and it originates right here in these cells. There's no other input to them. And so this is kind of right here in the cell, the magic of that heart beating. This is where it's coming from. And so if we want to know why, how does the heart do that? It's like, well, how do the cells do this? It's kind of stepping down one level. But the question's kind of the same. How does it do that?
So Cathy, maybe I'm jumping ahead of you here, so I'll wait if I am. But how does that work then? So it can do it on its own, but then how does it coordinate with the autonomic nervous system? Like when you exercise or sleep or whatever, and your heart rate changes. So it has to have some sort of coordination, I would imagine, with the nervous system.
So that's part of what we're interested in, and I'll show you some more of that in just a second. I'm not going to talk at length about that, but we're interested also in what happens in the single cells when they're stimulated by, say, the sympathetic nervous system, what changes within the cell. But I won't talk very much about that today. So what I want to say first, before I show you that part, is that these spontaneous action potentials, APs, arise due to a spontaneous depolarization of the membrane potential that occurs during diastole. So this is a bunch of jargon. Diastole is the time, the part of the cardiac cycle when the heart's relaxing before it contracts again. And depolarization means body membrane voltage here for the single cell. So it's the voltage across the membrane. The inside is relatively negative compared to the outside. And you can see that in this interval between spikes, the membrane potential creeps up more negative so that it becomes less polarized, is what the jargon means. And this creep in membrane potential, shown by this arrow, triggers the next action potential. So this part of the action potential is the part that's responsible kind of for the magic of like, why do they go? Why is the membrane potential not stable? And so we just keep whittling down it to look at mechanism, like how does the heart do that? Well, it's because of the cells. How do the cells do that? Well, it's because their membrane potential does this. Well, why does the membrane potential do that? So we keep just asking the same question in different ways, which is kind of job security. Because you're never going to figure it out. The fact that you look, did that make sense? Okay. Okay, so Rob, this is the answer to your question, partly. We can recapitulate right here, single cell, sympathetic nervous system, fight or flight, increase in heart rate, by washing on a beta receptor agonist. So you've heard of beta blockers, right? So this is a beta stimulating drug. It's called iso. And right here in this cell, same cell, we wash the stuff on it. It's the same cell, and now the action potentials are faster. Right? So the cell's going faster, the heart's going faster. And interestingly, if you look at most of the action potential, it's kind of the same, the black one and the blue one, about the same shape. And the difference is really all about this diastolic interval. Now the depolarization is faster, it's a steeper slope. That's what makes the next action potential come sooner. So once again, whatever is going on during this interval, what is really what's responsible both for making the heart beat and for then making it beat faster in response to sympathetic nervous system stimulation when you're scared or running.
So you're just making like discrete adjustments to that drug to see where is that change happening, and what do you have to do to keep manipulating that?
I'm not sure I understand. Let me tell you how we do the experiment and see if it makes sense, because I'm not sure we're connecting. Which is this is why this is really important, right? So we have these cells, they're isolated from the mouse heart, which I didn't mention, but the mouse heart is like little, of course, and then this is a little part of the little mouse heart, and then this is a single cell under there. So that part of it's kind of magic to So we put these cells in this chamber on the microscope, and there's fluid in the chamber. It's flowing through all the time. It's the cell in the dry environment will die all the time. So it has to have a saline solution washing over it. So then we just change to a saline solution that includes this drug, right? So it's not like locally applied. I think that was one of your question about like can you put it on one part of the cell or another. And that would be a really cool experiment. And maybe we could do some kind of complicated manipulations to do that. But looking at sort of subcellular things like that is more difficult. Any more questions? Okay, so we're interested in what's happening in this diastolic interval that makes the membrane potential default. Those words sort of make sense now. Okay. Well, I said this is membrane voltage, and so membrane. We have to do physics here. This is Ohm's law. my husband's an electrical engineer, so we have this similar kind of gap. Is like he speaks electronics and I speak cellular electrophysiology, and you would think we'd be able to talk to each other. So, but he likes Ohm's law, so that's kind of good. we can connect over Ohm's law, right? So what Ohm's law says is this relationship that voltage, which we're plotting here, this is what we're measuring, equals current denoted by an I, logically, times resistance, right? So the changes in membrane voltage in these cells come about because of changes. This is whittling down again. Come about because of changes in currents that are flowing. The currents across the cell membrane are carried by ion channels. So sodium and potassium and calcium ions are these charged particles that flow through these proteins across the membrane. They're carrying a current, and it changes the membrane voltage. Okay. So it's unfortunately necessary to whittle down.
And John Bankston in his seminar, he talked quite a bit
talked
about
ion channels.
the ion channels. Yeah,
Okay.
so we have a little, at least
So
those
you
of us who
you
attended
guys
that
have a
were a
little
little
background.
background.
That's right. Okay, so John and I can talk to each other about ion channels.
So how would you get the solution into the heart? Like is that
Into the heart itself. You mean the blood?
well, so you are stimulating it with that solution of adrogenic or whatever you're putting in your liquid to make it beat faster. How do you, like, is this a pill someone's going to take? Or are you going to drip it in there from a pacemaker?
Yeah, so that's a good question. So there's a couple things about it. This is a single cell. So we take the heart out of the mouse, we cut out the little part of the heart, and then we dissociate the single cells. So there's kind of no heart left in these experiments. We only have this cell.
Right.
But I'll show you in a little bit.
Okay.
We can apply the same drug to mice or humans, and so you can do it by injecting it. Is how we do these experiments. So it can be applied like a beta blocker can be taken as a pill, or it can be infused into an injection or whatever. So in principle, any drug that we wanted to develop could be applied and could have effects on these cells by eventually making it into the blood. And these cells are in close contact. Does that answer your question? Absolutely. Yeah, it's like these practical things. Yeah. Okay, so we'll do all that. Okay, so I said there's lots and lots of ionic currents that are important for pacemaking. And one of the controversies in the field, it's kind of ridiculous, and it's like, what current is the most important? And I'm going to come back to that point at the end. And so I like to stay out of that because they're all important, it turns out. But one that we focus on is called the funny current. I said I is for current, so this is abbreviated IF. And it's called funny because it has properties that are kind of backwards to many other currents. Sodium currents and potassium currents, and calcium currents are better behaved than these ones. So that the currents are produced by ion channels, and I drew this one. This is my artist rendition of this particular ion channel that conducts this current. And the reason we're interested in it is because it's highly expressed in the sinoatrial node, and that's what this thing is supposed to show you. You can label these channels, they're called HCM4 channels, with a green fluorescent marker. And in the sinoatrial node, there it is, it's all green. Here's a part of the heart that's right next to the sinoatrial node. There's no channel expression. So these channels are actually used as a marker of this part of the heart, and that's true in humans, it's true in mice, it's true in all animals. So we think they're important. I'm going to skip over this a little bit, and I'll come back to that a little bit later. And then the other reason we're interested in these particular channels and this particular current as one focus is that there's all these mutations. So this is another artist rendition of the ion channel. And all the red dots are mutations that have been found in different humans, different people who have sinoatrial node dysfunction. So if you mess up the channel with a mutation, you cause the sinoatrial node to do funny things, right? Inappropriate things. And we also know that if you block part of the current, the drug that I mentioned before, this drug called ivabradine, it can slow the heart rate. So this is from people, heart rate in humans over the course of several months of treatment of the drug, and their resting heart rate drops by 10 beats a minute or so. So for these reasons, we know that this current and the channels that produce it are critical for pacemaking, and that things that you do to the channels alter pacemaking, and the channels are highly expressed in the sinoatrial node. What also makes them an attractive drug target is they're not expressed in other parts of the heart. So there's all these channels and currents and everything. This thing go up and down, and it's not just one of them. But most of the other ones are also found in other parts of the heart, or they're found in the brain. So if you make a drug, then you have a lot of off-target effects, or you have a lot of side effects. So it's thought that by targeting something that's really highly localized to the sinoatrial node, it might make a drug that's more specific for targeting heart rate without off-target effects, without side effects. All right, so those are some of the reasons we focus on this particular current and this particular channel that produces it. And that's what I have for a bit of kind of background overview. And so kind of getting back a little to Rob's question, like, well, what about this, sympathetic stimulation of the heart? That's one of the questions we're interested in. Another question we're interested in is much more like what John Banks does, looking at the ion channel biophysics. So even within this molecule, how do different parts interact together? How do different proteins interact with these channels? So these are all kind of what you might think of as sort of bottom-up approaches. You start with like this thing. We know it's important. It's kind of a string you can pull on to start from a very small level, going back to the whole heart level. So that's one type of approach that we use in our experiments and our projects. I'm not going to talk about that today. And the other type of approach we use is we start at the whole animal level and sort of whittle down, kind of more what I've just been, I started with the heart and start whittling down. So I'm going to tell you just about one particular project we're working on that kind of, it shows you a bit of that approach and shows you a little bit of the details that we look at. Okay, so this whole category of more kind of top-down experiments, we look at how the sinoatrial node responds to physiological challenges, which is, I don't know what to call them. So we've looked the most so far at aging, and that's what I'm going to tell you about. And we'll also mention in passing that we have parallel projects that follow kind of a same type of experimental approach to look at how diabetes changes pacemaker activity and how exercise training changes pacemaker activity. So aging. These are data from humans again, people like us. showing this really kind of disturbing thing that one's maximum heart rate decreases with age. This is age, this is maximum heart rate. And here's the really depressing thing about it. There's actually 3 lines on here. There's sedentary people and active people and endurance trained athletes. The heart rate decreases at the same rate for everybody, which is it's kind of awful, right? So the decrease in heart rate and maximum heart rate is considered then to be, it's an inherent property of aging. It's independent of lifestyle, gender, physical fitness. It just goes downhill for everybody. Yeah, I know. So we're all hooked. And we care about maximum heart rate. And like how much, when's the last time I got my heart rate to max? It's like, it was uncomfortable and I stopped as quickly as possible. So, you can say, well, who cares, really? Most of the time we're not operating in maximum heart rate range, and unless you care about your 5K time or something, like, what, right? But it does matter, particularly for people over here, towards the right end of the scale, where we'll all get someday, we hope, right? That this age-dependent decrease in maximum heart rate contributes to a decrease in maximum oxygen consumption, VO2 max, with age. So there's a lot of things that affect VO2 max. And fortunately, if you exercise, you can train most of those other things to sort of compensate. But since maximum heart rate is part of this, if maximum heart rate goes down, there's an inherent decrease in VO2 max. And for people over here, if this said VO2 max here, there's kind of a lower line that you can draw. And if your VO2 max is below that line, you don't have sufficient aerobic capacity to bathe yourself, dress, live independently. So it contributes to a loss of functional independence for people. So one of the take-home messages here is like, well, we can't do anything about maximum heart rate, but we can, by physical exercise, train all these other things. So that's the secret to this. But many people maybe can exercise, and certainly many people don't. So that's part of the significance. So what causes? Here we go. We're just gonna well, that's interesting. Well, what makes the maximum heart rate go downhill with age? No matter what thing it is, we always just ask the next question.
Is it just that the cells just get?
Well, yes, and I'll show you that, and that's where we're going. But the next thing that's been known for a really long time, so these purple line data were published in the 1970s, so nearly 50 years ago, and the observation precedes even this publication too. So this is a lower heart rate. This is all on the same scale. This is maximum heart rate. So this is what maximum stimulation of the sympathetic nervous system, maximum fight or flight response, right? This is what's called intrinsic heart rate, and it's essentially at the in a whole life person that independent heartbeat. So to measure this in a human, or I'll show you mice in a few minutes, you block all of that sympathetic nervous activity with a drug, which you can infuse into a human. And you measure this intrinsic heart rate, which goes downhill with age, and it's completely parallel. So the slopes of these lines are the same. Okay, so what does that mean? The intrinsic heart rate is measured during autonomic nervous system blockade, and therefore this reflects at the whole human level, whole animal level, sinoatrial node activity. And if it goes downhill with age, therefore, that means that sinoatrial node activity is compromised with age. It's exactly what you just said, right? Okay, so we want to look at this in mice. Yeah.
So like if someone were to go home, work out, they're 30 years old, push their heart rate to 205, what is that?
Oh, yeah. What you can appreciate maybe more in this data set is that, each of these dots is an individual. Right? So there's a lot of individual variability, right? So these lines are true on average, but your maximum heart rate might be higher. It's still going to go downhill at about 1 beat per year, 1 beat per minute per year. Yeah. So it, yeah, but the absolute value for an individual can vary quite a bit off of that. And that's a really common question. Like, well, my heart rate is whatever. Right? Yeah. Well, people do ask that. That's a
So,
good question.
Cathy, like, we know that as we age, like, you lose hormones, you stop, your ovaries don't have eggs in them anymore. I mean, things change.
Things change
All
all wear
downhill
down.
from us.
Yeah, so, I mean, it seems like that would be true of, our working system. But I wonder if maybe some of those things that we also decrease in may be affecting the heart,
Yes,
It's
certainly true. Absolutely true. There's all these circulating factors. Heart rate is so important. Think of the evolutionary pressure. Like you see a tiger, right? Like so heart rate's highly regulated, and there's all these circulating factors, hormones, peptides, and whatever that affect heart rate. So that definitely contributes. But what I'm going to show you that in addition to that, there's something messed up with single cells too, and so we've focused in on that aspect of it. But it's not the only aspect, and there's other things too, like you have fewer pacemaker cells as you get older, and there's fibrosis in the heart. So this electrical signal that originates in the sinoatrial node doesn't spread as readily through the rest of the heart. So there's a lot of factors. All right, so we wanted to study what happens, and it's hard to do in humans. We can make humans exercise and use drugs, but you can't isolate sinoatrial nodes. So people don't want to give those up. So we use a mouse model. Rob was here when Eric Larson was in the lab, who was a graduate student. He had this really nice paper in high impact journal. It was really great. So Eric decided to take this aging question on, and he devised this way to measure heart rates from mice. So that's the little schematic here. And they're not anesthetized because anesthesia messes up heart rate. So there's little foot pad electrodes, and you put in this little tube, and then you measure these little mouse ECGs this way. And from that, that's what's shown up here. From that you can get heart rate. So what's plotted here are just mice of two different ages. We actually had some others, but the 2 to 3 month old mice are kind of more or less 17 to 20 years. So it's not a direct correlation. And then 21 to 24 month old mice are more or less 59 to 65 human years. We also had a much older age group. They were like 85-year-old mice. They were really, really frail. Yeah. But the thing is, it turns out for aging studies, if you want to isolate the variable of aging, then it's better to use less old age groups so you don't have all these other things that happen with aging too that could maybe have secondary effects on the thing you're measuring. So in the aging community, or in the aging literature, this is considered to be an age group where you can see the effects of aging kind of more or less in isolation from secondary effects. All right, so Eric measured their maximum heart rates in the mice, and the red bar is the old guys, and the black bar is the young guys, and there was a significant decrease in maximum heart rate, and
That's
also
the asterisk means.
that, yeah, thank you. So I just popped that word out there without thinking. So we do statistical analysis on this. So this is like a t-test here, and the asterisk represents a certainty level of at least 0.05. So it's less than a 5% chance that we got this wrong based on these data. And I didn't write down the actual p-value here. Looking at the data, it's probably quite a bit less than that for this particular thing. But that's usually the standard that awards the data set the asterisk as significant. Yeah. And then Eric also measured in the same mice their intrinsic heart rates. So he shot them up with these beta blockers, basically. And you can see that the intrinsic heart rates were also significantly slower in the mice, right? In the older mice. So in the mice, then, we can look at the single cells. So that's what he did. And now instead of measuring, is it like the parallel, completely parallel, slides and figures. But now these are action potentials. They're recorded from a single cell, just like I showed you before. And this is a schematic that I might or might not have time to get to, but how we actually record the action potentials. And once again, they're reversed from the last one actually. So the maximum action potential firing rate is significantly lower in the single cells than the old animals. And then when we take away the ISO, the basal action potential firing rate is just slow. So whatever's causing the heart rate to be slower, this must contribute. There's probably a bunch of other things too, but this must. And so we always ask the next question, well, why are they going slow? So that's what we decided to do next. And I said, Eric was a grad student. He wanted to graduate sometime. And I said, there's lots and lots of ionic currents in these cells. So why are the action potentials going slower? It's like, hey, Eric, how about if you spend the next 20 years like measuring every current in these cells? So that didn't seem very viable as an approach. Right? So we decided to be a little bit smart instead. And at least make some hypotheses, which can be right, they can be wrong, we don't care, but at least we have something to limit the possibilities. Otherwise, science is just like this endless description. So kind of the broader principle of a hypothesis is at least you make a little step forward in your knowledge, is like yes or no, right? Instead of like kind of testing the whole world. So to say, well, why are the old cells slower? We looked at the shape of the action potentials, which vary quite a lot. I see that here, but we lined them up sort of on average. And so here's an average action potential from the young cells in black, and the old cells are shown in red. And this is kind of opposite from what I showed you in the first place from the ISO. Is that, well, the action potential itself is kind of upstroke in the waveform. The peak of it kind of looks about the same. And the differences are due to this diastolic interval, which is now kind of depressed or slower in the cells from the older animals. Okay, so Eric did, in the first place, a really tedious manual analysis. He got all of these individual action potentials from all of the cells and tried to say how much slower is this little part than that little part, and lining up lines and stuff like that. So that also wasn't very efficient. And so I wanted to say that just because you guys know people in the lab, right? So Christian Rickert, you know him, so he's the German guy. Okay, so Chris wrote this little software program, and that's called ParamAP. So it automatically kind of gets all these little parameters out of the action potential, so we can quantify. We can see by eye that something's different. Is it significantly different? Which part of it's different? So we want to focus our hypothesis a bit, and this is a way we can do it. So this is what ParamAP, the software, does, and then we can line up all of the action potential waveform parameters. So there's time intervals, this little part and that little part. There's membrane potentials. There's the most negative potential and the peak potential. And then there's kind of rates of change in membrane potentials, some slopes and lines. So we can line all of these up and compare young and old and do it all on average. And the way to read this is that the red ones are statistically significantly different in old cells versus young cells. So most of the parameters, just like you see by eye, are not different, but the ones that are associated with the diastolic interval are different. The diastolic depolarization and the most negative potential than the maximum diastolic potential. So So this kind of approach, again, it sort of restricts what it is we have to look at. And so I'm going to just kind of cut to the punchline on this and say that these parameters happen to be associated with our favorite current, IF. We looked at that subsequently, and that is also changed. And so, again, we can say that must contribute to the slower firing rate in the older cells, which must contribute to the slower heart rate in the older animals. So I'll show you a few of those data, but I'm not going to go through it. What I wanted to do, though, and what I kind of mentioned, is that we have parallel experiments going on in aging, diabetes, and exercise training. And so I'll mention those kind of briefly here. So aging is red, diabetes is blue, and exercise training is green. So what's plotted here is maximum heart rate compared to young, healthy, controlled, non-diabetic, and sedentary mice. Right, so those are the controls. So the maximum heart rate is slower in the old mice. The maximum heart rate is also slower in the diabetic mice, and the STZ stands for this drug that kills off their pancreatic beta cells and makes them diabetic. And the maximum heart rate, fortunately, is not actually changed in the animals that were exercised. And that exercise was kind of fun. It was swim training. Well, it's kind of fun in principle. The actual doing of it was pretty tedious. So it was 2 hours a day of swimming, and the mice are in this tank. So they get 1 hour in the morning and 1 hour at night. You throw the mice in the water. And then we hired some undergrads from Boulder to sit there for the hour. You know, they're mostly on their phone, and then they'd be like, "Hey, sit."
No holding on to the side.
Yeah, or they'd hold on to each other,
Drown the other one.
Yeah, and they got to know the mice after a while. I was like, "Oh, that guy, cheater, right?" You'd find like a little crack.
They come with floaties on.
I have to say, it made for one of the most interesting expense reports we ever processed. When Rebecca was here.
Swimming pools.
Yeah, we're like, swimming tanks for mice? What the heck?
And we had to get some fish tank pumps so they couldn't just float, right? So the fish tank pumps make the water move, and we had to get fish tank heaters because they have to, you know, for Ayacook, they have to be all warm, and you have to take them out and dry them and everything.
So these are all allowable costs.
Yes.
Yes. Reasonable.
I should have put it in here because Dan was a guy who did the exercise experiments, and he made some movies of them, and they was like kind of swimming around. All right, so that was the
Maybe swim
maximum heart
caps.
rate.
So Kathy,
Yeah.
the red, the blue, and the green, are those young mice?
No.
No.
So
They're all old.
the young guys are black. They're just young. They don't have
Red or old.
diabetes, and they weren't exercised.
Okay.
The red ones are old ones.
Okay.
The blue ones are young ones with diabetes.
Okay, young ones.
And the green ones are young ones with exercise training.
Okay.
And they're all kind of compared to their
the
own
old.
controls, right?
Okay.
And then we do the same thing with intrinsic heart rate. And what's interesting is that in the old mice, in the diabetic mice, and in the exercise trained mice too, the intrinsic heart rate is significantly lower than in the controls. And so this thing about exercise training, it seems like you're kind of like, how does that make sense?
Well, of course, you think of like a professional athlete who has a very low resting heart rate. Is that a similar, same effect?
Well,
you're
this is
seeing?
gonna contribute to the resting heart rate. So for so the resting heart rate's kind of complicated because, you know, what's rest, right? Is this rest or is this rest or is that right? So the resting heart rate is finely tuned by by the autonomic nervous system, either up or down. So this definitely contributes to the slower resting heart rate, but that's a little bit more of a complicated question, actually, right? Resting heart rate always confuses me. Okay, so these things, at least for the intrinsic heart rate, look to be sort of the same phenotype. And aging and diabetes, like both the maximum heart rate and intrinsic heart rate, are decreased, right? And it's been written that diabetes might be sort of an accelerated form of aging, blah, blah, blah. So what we did is the same thing. So we made these complicated graphs, but what you can look at are these action potential waveforms. And the thing is, I said that the action potential waveforms give you some hints about what's going on, the underlying currents. And the punchline, the take-home message, is that even though some of these phenotypes seem really similar at the whole animal heart rate level, at the single cell level, they're quite different. So probably, at least for sinoatrial node myocytes, aging and diabetes are not the same. And one would fear that they're additive, although we haven't looked at that yet, but it's like if you're old and diabetic, that could be a sort of a double whammy. And then exercise training, we're going to assume is beneficial, right? And it's making like a host of changes in all different action potential waveforms and presumably underlying currents that presumably all kind of balance out and work in a coordinated fashion. So that was kind of one of the take-home messages I wanted to show you. And I could go on a little bit, or we could just that's kind of a good stopping point.
Yeah, several of us have another meeting right at noon. So
Yeah, okay.
that
I
be a good stopping point.
didn't want to go over. So that's a bit of what we're working on, and thanks for listening.
Wow.
Mm-hmm.
Thank you.
I can tell the most important slide, though, is the one at the very end. Get to, because you guys know. Well, some of you guys know the people in the lab. And so, Em, who just graduated, she worked on the next aspect of the aging project. That was a big part of her thesis work. And Hisham, he's doing kind of more molecular work. You've seen him around. And Chris, I showed you, he's got the programming thing and he's doing another project. And Dan was doing the exercise training project.
Very
And all the
cool.
funding and everything. Yeah. Okay, so thanks.
Yeah.
Thanks, you guys.
Fascinating work, Cathy. Thank you. Any last questions?